Okay, so welcome everybody to our genetic testing session. Does anybody have any questions from prior sessions? Any questions? Today we're going to talk about genetic testing. We'll introduce the topic of next generation sequencing today, but next week we're going to devote the whole session to next generation sequencing, and our scenario next week is going to be you have a patient who comes in with a condition and you do next generation sequencing as a means of variant discovery, trying to find out exactly what variant might be causing the phenotype. Then we'll cover genetic engineering, clinical genetics, and then we'll have our ethics debate on the 20th. Okay, so sorry we had to skip last week. We had toyed with the idea of introducing a pharmacogenomics module. I don't think we're going to work that in this cycle. Instead, today we're going to spend a little bit of the genetic testing time talking about pharmacogenomics, and we'll have a case, a pharmacogenomics case, and we'll look at some of the bioinformatics aspects of pharmacogenomics. So that'll be it for pharmacogenomics, is what we cover today. And then I think the next time we go through this series, we're going to try to introduce a whole module just devoted to that particular topic. So for today, what we're going to do is first introduce the notion of genetic testing, and we're going to shift the case after our discussion of types of tests, because what I'd like to do today is walk through the case of a patient who we're going to prescribe a drug for which there's a genotype interaction, and as a class kind of think through what kind of test would we like to do. And so we'll go through all the different types of tests, and then in addition to the typical quiz questions that we weave in, we'll specifically be looking at this case in the context of genetic testing. Okay? So any other questions before we dig in? We've got a couple of minutes. We can go over questions if there's any. Okay. No questions. So, a lot of you guys on the call are clinicians, and this slide may be pretty obvious, but we anticipate that there will be students that have less of a clinical background, and so we wanted to kind of bring home the point that when you look at the mountain of genetic tests that are available, you really need to think about why you're ordering a test in order to know what test to order. And I know I say that and it's like, well, duh, for the clinicians on the call, but generally, at a very high level, what will happen in a clinical workflow is a clinician will see a patient, they'll obtain family history, they'll obtain clinical data such as lab data, they'll obtain phenotype information such as a physical exam, and based on that, there'll be a suspicion about some type of disease, and a test will be ordered in order to, for instance, confirm that diagnosis. And there's such a range of tests, as we'll talk about, ranging from cytogenetic tests, where you're looking at DNA or chromosomes under a microscope, to molecular tests such as next-generation sequencing, There are targeted tests where you're looking for a very specific mutation in a specific gene, ranging to whole genome testing. There's a number of considerations in this, and really it's impossible to know what test to do if you don't know why you're doing a test. And so we're going to cover all of these tests, and then we're going to come back with a specific case of a patient about to be prescribed a drug, abacavir, that has a known interaction with a particular genotype. And we're going to say, okay, now that we understand the spectrum of tests, which test specifically do we want to order for this? And I guess the other reason I showed this slide is to kind of dispel the notion that all we need to do is whole genome sequencing on everyone. I'll partly dispel that notion today. I'll far more strongly dispel that notion next week when we really get into some of the caveats and nuances and strengths and weaknesses of whole genome sequencing. When you have performed a test, depending on the test that you've performed, you may encounter several things. You may encounter variants of unknown significance. For instance, when you consider that a typical person has roughly 4 million variants when compared to a reference genome, the vast majority of those 4 million variants are of unknown clinical significance at this point. So honing in on which of those variants might actually be the causative variant has been the subject of quite a bit of study, and guidelines have been written about it. But managing all of these other variants of unknown significance is a big consideration when you're dealing with particularly things like whole genome testing. Other findings crop up, for instance, if you're doing whole genome testing, because you may have ordered a test to look for X, but you may have discovered a mutation that is, highly correlated with a particular disease phenotype. Sensitivity and specificity, in a lot of ways, are similar in concept to sensitivity and specificity of any lab test. When you're doing a genetic test, it helps to understand the method being used to know exactly what that test can detect and what that test will not be detecting. For instance, if you're doing a targeted test where you're just looking within a gene for 3 specific mutations, well, there might be a novel mutation in that gene, and the test is not designed to pick that up, so it's just not going to report. It's going to report that the mutations it was looking for weren't present. You may have a test that reports a novel variant, but the question is that novel variant pathogenic? So sensitivity and specificity issues are kind of sort of similar to other lab tests, but also there's some nuances in that each one of these tests is designed to focus on a specific aspect, specific genes, for instance. And so if the results come back negative, it's important to understand exactly what the test was looking for, because then, for instance, if there's clinical suspicion, you may need to do further testing. And then finally, once the test has been performed and interpreted, then we have this notion of treatment and follow-up, where we may apply precision medicine principles like we talked about in the cancer genetic session last time. Pregnancy risk, what are the risks to the offspring? And screening, if you have a particular mutation, perhaps you're going to need more frequent, colorectal cancer screening, for instance. So this is the framework by which we're going to be looking at testing today. And primarily, we're going to be looking at testing from the context of a clinician who's assessed a patient, is suspecting a disease, and then is trying to do a confirmatory test, and they're trying to figure out what type of confirmatory test might be indicated.
Bob?
Yeah.
Jonathan, can I make a comment for non-clinicians out there? In
Yeah.
any testing in medicine, it's far more complex than it seems. You know, you go to your doctor for a physical and you get a bunch of routine screening tests. Well, it turns out, as I'm sure you know, we have to think about what tests we're going to order, because if tests have a range that's normal and 5% are high normal and 5% are low normal, and you screen someone who's unlikely to have an illness, you're going to get 10% of the tests being abnormal, high or low. You really want to focus on trying to pick up the people where there be some a priori pre-existing risks, so that you're not getting lots of these people with false positives and false negatives. It's more meaningful to get a significant result. And this has some implications, as you were saying. You're doing genetic testing, but you aren't quite sure what you're looking for. You're going to get lots of information, and a lot of maybe not that helpful or worry you unnecessarily. I think there are some other things that come in genetic testing, but in all of medicine, we kind of have to think, what information do we want, and how useful will it be, and what will it lead to? I think everybody's heard breast cancer screening done too young leads to a lot of inappropriate, unnecessary biopsies and worry and concerns. It can become the same kind of thing. But an area that's going to get very interesting in genetics, as you have brought up, is, well, what if you have this gene, but it can be changed by your environment, by your age, and that's another whole area that we don't even know how to test.
Yeah.
We have some basic things to test for, but we're not going to, it seems like some things, like sensitivity to gluten, may change with time, and it may not even be able to be pre-tested and pre-screened. So there'll be a lot of interesting things, but thinking about why you're ordering something and what the chance of finding something may have some meaning.
Yeah, and in fact, it's a really good point, and I think we're going to come back to at least portions of your comment, Jonathan, when we talk about the clinical informatics scenario, because, assume, for instance, you've done whole genome testing on a patient, and now you have all this data sitting there. Should you check it every year based on evolving evidence? So it's a good comment that you made, and it's an important theme, and we're going to be revisiting that in a couple of sessions from now. This picture here tries to give you a super high-level view of the scope of genetic testing, and it does it in a way based on increasing resolution. So, for instance, karyotyping is a test where you're looking at chromosomes under the microscope, and obviously, when you're looking at a chromosome under the microscope, you're not going to be able to see a single base pair mutation, and so part of the determination about what test you need to order has to do with, well, at what size are you looking for? If you're looking for trisomy 21, then generally karyotyping works just fine. But if you're looking for a point mutation, then you need to order a test that's a molecular test that's much more fine-grained. It gives you resolution down to the 1 to 1,000 base pair range. So up here at the super low resolution, where you're looking at the entire genome, we have karyotyping, and so we'll look at karyotyping. You get into this kind of intermediate range where we have DNA chip technology or chromosome microarrays. You have this other technology, FISH, which we're going to look at, which is kind of like somewhere in between its resolution capabilities between standard karyotyping and chromosomal microarrays. But really then, if you want to get down to the super fine-grained units of resolution, the nucleotide level of resolution, then it's down to whole genome sequencing, or for targeted areas of the genome, if you're looking for particular point mutations, there are these things called oligonucleotide probes, where you have a small piece of DNA that's either radio-labeled or fluorescent-labeled, and you incubate it with the subject's DNA. And if it sticks, then you assume that it's complementary to the subject's DNA. So we'll look at these oligonucleotide probes, and what we're going to kind of do in the slides that follow is we're going to start here at the kind of microscopic view and work our way down to the molecular view. So we'll start with karyotyping, and probably most people are familiar with karyotyping. It's been around for a long, long time. It's a process where you take the metaphase nucleus and you stain the chromosomes, and then you look at them under the microscope. And when you look at the chromosomes under a microscope, you'll see that there are characteristic bands that form. The chromosomes 1 through 22 are simply numbered based on size. And so chromosome 1 is the biggest, chromosome 22 is the smallest. Chromosomes have a centromere. In some cases, the centromere is in the middle of the chromosome. In some cases, the centromere is towards one end, and in some cases, the centromere is almost completely near the tip. The short arm of the chromosome is P for petite, and then the long arm is Q. And so oftentimes, based on karyotype, and we'll talk about, we saw something on chromosome 1, P31, which would be this band here, or chromosome 7, Q31, which would be here, or chromosome 22, Q22, which would be chromosome 22, Q22, which would be way down here at the tip. And so we take the chromosomes and we stain them, and we get these characteristic bands. And these bands can then be lined up so that you can see this, for instance, for chromosome 1 would be the chromosome that you inherited from one of your parents, and then this would be the chromosome that was inherited from the other parent. And you can line them all up, and this is obviously a very gross view. With something like this, you could determine if there's a trisomy, you could determine if there's a monosomy. You might be able to detect large translocations, particularly because people that do karyotyping become very familiar with the banding pattern. So if there was a large translocation, you'd be able to see it. If there was a large inversion, you'd be able to see it grossly like this. And then, of course, as things get smaller and smaller, obviously you wouldn't be able to see it on a karyotype anymore. There have been some efforts to improve the resolution of the karyotype, and this is in part historic. I don't really know enough to say whether or not people are still doing these tests and at what frequency, but karyotyping predated fish technology, and fish technology predated chip technology, and chip technology really predated next generation sequencing technology. And so early on, there were attempts to increase the resolution of the karyotype. And when you go and typically when you're doing a karyotype, you're looking at a metaphase chromosome. And if you remember mitosis, you'll remember that the chromosome progressively condenses as it goes from prophase to metaphase, right before you have anaphase and then the separation. So here we see the chromosome that's in prophase, and it gradually condenses to a chromosome here that's in prometaphase, and then it condenses to the chromosome in metaphase. And in metaphase, when the chromosome is stained, you can see, depending on the stain you use, somewhere around 300 to 450 bands across all the chromosomes, whereas if you apply that stain to a prophase chromosome, you can see about 800 bands. It's a little bit more complicated to do the procedure. I don't think it's very commonly done anymore, but it was an early attempt to kind of increase the resolution to get more out of a karyotype. This would allow you to see, inversions, deletions, translocations that you wouldn't necessarily have picked up on a standard karyotype. So that evolved to a technique called FISH, or fluorescence in situ hybridization. And in this technique, what you're doing is you still have your chromosome laid out on the slide, but now you have a large probe. And this probe is labeled with fluorescent dye and is allowed to hybridize with the chromosome. And now you're looking at the chromosome under the microscope, but you're looking for areas where the chromosome fluoresces. And these fluorescent probes don't need to be anywhere near as large as a band in a karyotype. And as a result, the resolution of FISH is far above the resolution that you can obtain with a karyotype. So here, for instance, we see a FISH technology. These red dots are, and just a reminder that there's a lot of details about the pictures in the notes in the slides in terms of exactly what probes are being used and things like that. So in this particular picture, we see a probe. This is for 4Q12, and the green probe is for 4Q31.1. And as expected, when you have a fluorescently labeled probe for 4Q12, you would expect there to be two red spots because there's going to be two chromosome number 4s. So just as expected, you see the two red spots, and let's see, can you see them over here? Here's one here, and I don't see the other one. Oh, here's the other one over here. And then the green spots are for 4Q31.1, but you can see that these probes allow you to target an analysis of a specific region of a chromosome with much greater resolution than you could obtain with a karyotype. Down here on the bottom, the blue probe is for chromosome 18 centromere. The green probe is for the chromosome X centromere, and the red probe is for the chromosome Y centromere. Okay. So who can tell me what sex this person is?
Male.
It's a male, right? Because it has a probe. It has a probe to the X chromosome that found a match and hybridized, and it has a probe to the Y chromosome that found a match and hybridized. Had this been a female, you'd see two green spots and no red spot. Okay. Had this been a patient with Turner syndrome, for instance, who's just XO, just one X chromosome, and no other X and no other Y, you would see one green dot, and that's it. If this had been a patient, for instance, with Klinefelter syndrome that has two X's and one Y, you'd see two green dots and one red dot. Okay? Here, this is FISH technology. The thin arrow is a probe for the chromosome 17 centromere. And we see that there's two of those. So we know that there's two chromosome 17s. The thick arrow is for a portion of 17p. It's for the short arm of 17p. There's a partial deletion of 17p, and this patient clinically had a syndrome, Smith-Magenis syndrome. And so here you can see that we only have a single probe for 17p. There's also multicolor fish, where the probes for different chromosomes are designed to have different fluorescent colors. And that allows you to do something like a fancy schmancy karyotype, a spectral karyotype, if you will, with fish. And in this particular case, you can see if you look at chromosome 6 and you look at chromosome 16, you'll see that there's been a translocation. This yellow tip here came from chromosome 16, and in exchange, chromosome 16 now has a tip that it obtained from chromosome 6. There's also a fairly complex structural abnormality between chromosomes 2 and 10, where the portion of chromosome 10 looks like it was derived from chromosome 2, and then the part of chromosome 10 that was contributed in exchange back to chromosome 2 has a break in it, as if not only was there a translocation, but there was a subsequent inversion of this particular segment. Some of these changes may be gross enough that they would show up on a karyotype or an enhanced prophase karyotype. But with the FISH technology, the probes can be far smaller than what you would typically be able to see under the microscope with a karyotype. And that was the strength of FISH. Although FISH technology is now also kind of fading, because when you look at even these large structural abnormalities, many of these can now be inferred based on chip technology or based on complete DNA sequencing technology. Okay. So far, so good? Okay. Kind of tricky stuff, but I think it kind of makes sense, right? You're staining the chromosomes, and it's pretty interesting when you think about, you know, how did they design the probes, and and most of these probes that they use now are actually, you can figure out exactly what probes were used. There's commercial libraries of probes available, and we'll come back to this a little bit, because sometimes when a lab reports the results of a FISH test, they'll report exactly what probes were used, and that can be helpful information to help interpret the clinical significance of the results. So let's now talk about DNA chips, okay? Before we do, you know, we've talked about FISH probes, and in some ways the picture here is along the same lines of how a FISH probe is working, in that you start off with some type of probe, and that probe is designed to hybridize with an area of interest of DNA. Okay, so you have some kind of probe. It can be a radioactive probe, it can be a fluorescently labeled probe, and you design your probe based on where the area of DNA where you're going to be looking. The probe is mixed with the DNA. The probe hybridizes, and then you can detect whether or not DNA hybridized to the probe. So in a chip, imagine, if you will, you have a whole bunch of probes. You have a whole bunch of wells on a chip, and in each well, you've glued in a particular probe. Now you go ahead and chop up some DNA, throw it into these different wells, and there'll be some wells where the DNA will stick. And in those wells where the DNA sticks, you can infer that the DNA had a sequence that was complementary to the sequence of the probe. And that's kind of the basis of how all of these chips work. So first off, you have to figure out what it is you're probing for. There's really no such thing as just a generic chip. There are chips that look at all of the different known point mutations of a particular gene. There might be a chip that looks for all of the different mutations in the genome. But at some point, the chip maker has to decide what the purpose of the chip is. Based on that, they have to create a library of oligonucleotides. They then have to apply those oligonucleotides onto the chip. So at this particular position, we know that we have oligonucleotide 1. And here we have oligonucleotide 2. And we need to know exactly which oligonucleotide is in what position, because ultimately, after we've hybridized the chip with the patient's DNA, we're going to see where there was hybridization, because we're going to see a fluorescent label. And so based on the results here, we can infer, well, the sample DNA must have had a region that was complementary to the oligonucleotide that was placed in well number 1, because it hybridized to it, and therefore we're seeing a fluorescent spot. Okay, and so that's kind of the basis of chip technology. Is the chip maker defines oligonucleotides. Those oligonucleotides are attached to known positions on the chip. The chip is then hybridized with the sample, and then we read the sample to see in which wells there was hybridization. This slide, I think, gets a little bit confusing, and it might be a little bit over the top, but I bring it up just in case you have seen and were confused by the different types of chips that are out there. And there's lots of different types of chips out there, but the main types of chips used for clinical testing are based on either SNP arrays or comparative genome hybridization arrays. And I got a slide coming up for each one of these to try to better explain it. But at a high level, think of a SNP chip as having a bunch of little probes, where each probe is specific for a particular point mutation. So at position 3 in a particular gene, do you have an A in that spot, or do you have a G in that spot? And so one well would have a probe that would hybridize to DNA that has an A, and another well on that chip would have a probe that would hybridize to DNA having a G at that spot. And so you put both of those probes on the chip, you add the patient's DNA, and if the well, with the A lights up, now we know that a particular patient has an A at that particular spot, and so that particular SNP allele is an A as opposed to a G. And so the SNP arrays are probing for point mutations. These are generally small probes. It's really interesting, the hybridization technology. The conditions have to be adjusted so that even though this probe is several thousand base pairs long, you adjust the experimental conditions so that the DNA has to precisely be complementary to it in order to bind. Otherwise, it'll wash off. So these are small probes. The hybridization works when you have a complete complementary sequence. But as you can imagine, because they're small probes and you can only put so many probes on a particular slide, overall you're assessing less than 1% of the genome. You may only be assessing 1 million spots on the genome. Alternatively, you can do this technique called comparative genome hybridization, where you're really probing for the presence of segments. And so here you have a probe that can be up to 150,000 kilobases. That's a big probe. That's not going to be sensitive to individual variations in SNPs. If this probe finds a corresponding segment of DNA, it's going to hybridize to it, and it will be tolerant of 1 or 2 SNP differences. And so these probes can be quite useful looking for the presence of segments. They can be quite useful looking for large segments of DNA that are missing. They can be quite useful looking for microdeletions or deletions in DNA that were below the level of resolution of FISH technology, far below the level of resolution of karyotyping. And in fact, as comparative genome hybridization first became a real technology, a whole boatload of microdeletion syndromes that previously the genetics weren't known began to be discovered. Large coverage, you can have up to 25% of the genome covered, 25% of the genome covered, because you're really looking for the presence of segments. Even when you're looking for point mutations, however, you can glean whether or not there are duplications or deletions, and it's kind of interesting the way that you would infer that. But imagine, if you will, a large segment of DNA where all you see is complete homozygosity. There's no heterozygosity in that complete segment. All of the SNPs you see are homozygous. You can infer from there that there could be a deletion of the other chromosome. And as a result, both techniques are complementary, and both techniques can be used to determine copy number variants, duplications, deletions, unbalanced translocations, and aneuploidies, but they're not suited for balanced translocations and inversions because, as you can imagine, all of the genetic material is still there. And so you're not going to see, and when you cut up the DNA, when you cut up the DNA and apply it to these wells, if it's a balanced translocation, you're still going to see everything, and if it's an inversion, you're still going to see anything. And so this also kind of helps realize, if what you're looking for is a particular large inversion or a particular translocation, then you have to decide, well, this chip technology, what I want to do, maybe I would still use FISH technology, or maybe I would still do a karyotype, if I'm looking for a particular condition. So there's 2 main types of these DNA chips. There's really a boatload of different types of DNA chips, but for clinical use, there's 2 main ones. It's the SNP array, where I'm adding these small probes, and if the DNA hybridizes to this probe, then I can infer that the patient must have had a T at this locus, and because this is the patient's DNA is labeled, it's going to light up the cell. And if there happens to be a segment of the patient's DNA that has a C, then that particular cell is going to light up. And I can put all kinds of probes on this slide. I'm not sure what the current capacity of these slides are, but it's, ballpark, you can put 1 million probes on a slide. So that would be a SNP array. And these other arrays, the comparative genome hybridization arrays, these are the larger probes. And what's happening here is, so here you have the microarray, and you have the large probes put in there. And you put in the patient's DNA, but you also put in reference DNA. And from there, you can glean, well, if the patient had an extra copy of a particular segment of their DNA, there's going to be more heavy binding, more heavy hybridizing to these particular wells. Whereas if the patient is missing a segment of their DNA, then the reference DNA is going to bind, and you're not going to see binding from the patient. And so there's going to be, a gain might be green, a loss might be red. And based on that, you can determine along the entire chromosome where the ratio of the patient's DNA binding to the reference DNA binding is sufficient in order to call either a duplication or deletion. And here what you see on this particular slide is a duplication of this particular region of the chromosome where there's heavy hybridization from the patient sample, much greater than what you're seeing from the reference DNA sample. And from there you can infer that there is an extra copy of this particular segment. Okay. These arrays are kind of tricky. You know, the basic principle is pretty straightforward. You're putting in probes, and then you apply patient DNA. Sometimes you apply patient and reference DNA. The inferencing that can be done based on these, I think, is where the kind of like the magic takes place. You know, it's very interesting to see what people can infer based on this. But in principle, what you're doing here is, you know, you're putting in some probes, and then you're putting in patient DNA, and that's allowing you to make all kinds of inferences about presence of SNPs, about the presence of duplications, deletions, etc. There's another type of chip. We talked about this a couple of weeks ago in the cancer genetics class, and this is an RNA expression array. And in this case, actually, we're not looking specifically at the patient's DNA, but rather we take the messenger RNA out of the cell. We reverse engineer the messenger RNA into complementary DNA. The probes in the wells are probes directed against the messenger RNA, and we're looking for hybridization. And then we can, for instance, look at a cancer cell and a normal cell, or we can look at a normal cell today and look at a normal cell under particular types of stress. And that can then give us an idea of what genes are overexpressing or what genes are underexpressing between a cancer cell and a normal cell, or between particular stress conditions. We saw this last week, for instance, in the cancer genetics, where we talked about precision medicine and showed an example where RNA expression array data is actually being used now to help guide the duration of hormone therapy in certain patients with breast cancer, by knowing which genes are overly active and which genes are underactive. So that is chip technology. Any questions about chip technology? Okay, you guys still out there?
Live.
Okay. Huh? Still there? Okay, just checking. Just checking. I don't want to put you guys to sleep. Okay. All right. Let's turn now to another test, PCR product size. And this is an interesting test because it's what's used for paternity testing and for DNA profiling. And so I thought folks might be interested to know how this particular type of test works. There's been, I think, 3 sessions where we've talked about these things called short tandem repeats. You may recall that your DNA is roughly 50% single copy and roughly 50% repetitive DNA. And when you look at the repetitive DNA, you may recall that there's a lot of different types of repeats. There's short repeats that are right next to each other, or so-called short tandem repeats. There are long tandem repeats. There are long dispersed repeats. There's a whole bunch of different kinds of repeats. But these short tandem repeats tend to be very interesting. in addition to discussing short tandem repeats, when we talked about genetic polymorphisms back in Deck 4, you may recall we also talked about short tandem repeats when we talked about Huntington's disease in Deck 5, where we talked about autosomal dominant inheritance with anticipation. And what's going on in patients with Huntington's disease is that when this chromosome is replicated, the number of repeats increases. And once the number of repeats increases past a critical juncture, the patient can then develop Huntington's disease. And in the last section, you may remember we talked about these repeats because in cancer, you can develop something called microsatellite instability. And microsatellite instability is where you begin to see quite a number of replications resulting in changes to the number of repeats at a particular short tandem repeat spot. Now there's thousands of these short tandem repeats scattered across the genome, and they have considerable polymorphism. So at a particular spot, I might have one of my chromosomes has 5 repeats and 3 repeats, whereas you might have at the same spot 7 repeats on one chromosome and 2 repeats on your other chromosome. Somebody else might have 9 repeats and 12 repeats. And so there are thousands of these short tandem repeats, and the short tandem repeat spots tend to have significant polymorphisms. So if you look at enough spots and say, okay, at this spot I have a 7 and a 2, at this spot, at this other locus, I have 12 repeats and 4 repeats. At this other spot, I might have 11 repeats and 3 repeats. If you look at enough spots, the probability of finding 2 people that have exactly the same number of repeats at all of those spots becomes astronomically small. And that's the basis of paternity testing, and that's the basis of DNA profiling, is that you look at a number of spots, 13 to 20 spots, and you simply see how many repeats you have at each one of those spots, and that becomes, if you will, your DNA fingerprint. So let's take a closer look at how this process actually works. But before getting into that, I found this kind of fun little slide because I know that there's a lot of terminologists on the call, and there's so much variability in the terminology around microsatellites and short tandem repeats, and you'll see that the definitions differ very widely, but I thought I would at least make you aware of this site, the Sequence Ontology site. And at least in HL7, when we debate definitions on the clinical genomics group, we typically defer to the Sequence Ontology as our source of truth. So you'll certainly see lots of definitions about microsatellites. Is a microsatellite less than 5 bases? Is it less than 10 bases? Is it synonymous with short tandem repeat? But here I'm showing you the definitions taken from the Sequence Ontology. And the Sequence Ontology is a very nice resource when the vocabulary that you're looking for really is DNA specific. It's not going to have like disease codes in there. It's going to have, DNA type terms in there. So satellite DNA is the general term for short sequences in tandem. About 10% of our DNA is satellite DNA. And we'll often break it down. There's other types of satellite DNA. Centromeres, for instance, are a form of satellite, but the repeats in the centromere satellites are more on the order of 150 bases. So microsatellites are also known as short tandem repeats, and that's what we're going to be focusing on here. These can be dinucleotide repeats. 2, 3, or 4. And then mini satellites, which people have also looked at using for instance, for paternity testing. Mini satellites are a little bit bigger for sequence ontology, 10 to 40 bases. Okay. So I thought what might be interesting also is to kind of revisit polymerase chain reaction, because here what we need to do is what we're going to wind up doing is cutting this fragment out. Okay, so we're going to cut here, and we're going to cut here. We're going to pull this fragment out. We're going to amplify it. We have to amplify it so that there's enough copies of it so that it shows up when we do, for instance, a gel electrophoresis. And so I want to go through just a little bit of the details about how do we isolate this particular region of the DNA, and how do we amplify it so that we can then infer what the repeat numbers are at a particular spot. So the way we isolate a particular segment of DNA is through, well, at least one of the ways we can isolate a particular segment of DNA. So again, what we want to do is isolate that particular segment and then amplify it so that when we run it through electrophoresis, there's enough of it to show up. What we can do here is apply this technique called PCR, or polymerase chain reaction, where what you're doing is, here's the target DNA, and right in the middle here is the short tandem repeat that we're interested in. So what we can do is we can add a primer to each of the strands. And we design the primer. These primers are also these oligonucleotides, 15 to 20 bases long. And we design the primers so that they stick right adjacent to the region of DNA that we're interested in. Okay, so we put in the primer, and then in the bath with the primer, we also have the DNA polymerase enzyme, and we have the free DNA nucleotides. And so this strand now grows, and this strand now grows. And you've just taken one strand that you're interested in, and you've just doubled it. We can cycle this now. We can now denature both of these strands, add new primers, and complete the process. And that's exactly what polymerase chain reaction is doing. It's running all of this through these repeated cycles where it heats it up, it adds the primer, it does primer extension, and then it starts the cycle all over again, so that in the end you wind up having millions of copies of the specific segment of DNA that you're interested in. Okay? Having done that, now that you have millions of copies of the particular segment of DNA that you're interested in, you can separate that DNA through a process such as gel electrophoresis. And in gel electrophoresis, you're putting your DNA samples up here and you're applying an electric current. And it turns out that for DNA, its charge is proportional to its size. And therefore, differences in migration rate correspond to differences in the number of bases in electrophoresed fragment. So if you have a small fragment that's only 4 bases long versus a fragment that's 5 bases long, that will have a proportionate effect on its charge. And so when you apply the current, the smaller the fragment, the more rapidly it's going to move towards the positive node. So small fragments will move quickly, and large fragments will move slowly. And so you can separate these fragments based on their size. So, if you're looking at a particular STR locus, and one person has 15 repeats, and another person has 4 repeats, well, the amplified fragment that just has 4 repeats is going to electrophorease very quickly, whereas the fragment having 15 tandem repeats is going to migrate more slowly because it's bigger. So let's look at that again. Here we have two alleles, and let's say that allele 1 has 4 repeats, whereas allele 2 has 8 repeats. We amplify these regions using PCR, and then we take the sample and we subject it to electrophoresis. And smaller fragments will migrate more rapidly, larger fragments will migrate more slowly, and so by electrophoresis, we've separated the two samples. And it's not just qualitative. It's not just, oh, we've separated the samples. It's also quantitative if you compare this to a reference sample. So if you've also, in parallel, done electrophoresis with a sample that has 4 repeats, and with a reference sample that has 8 repeats, you'll be able to say, allele 1, we know allele 1 has 4 repeats, and we know that allele 2 has 8 repeats. And when you do that, you wind up, and you do that at a number of different loci, you wind up then forming the basis for your paternity testing. So here, this particular portion of the slide on the left is electrophoresis for just one locus. And you can see here, patient 1 has alleles of 2 sizes. Patient 2 has alleles of 2 different sizes, and the 2 and 3 match. And so this might be an indication that these are the same people. But normally these tests can't just rely on a match at one locus, because just statistically speaking, there may be a match. And if you look at multiple loci, then the statistical power of the test goes way, way up, so that you can detect a match with a much higher probability. And so here you see on the right what's called an electropherogram, where the peaks on the graph represent alleles at loci. So individual 1 has an allele that has 325 repeats. He has an allele that has 320 repeats, etc. And the probability of anyone having the same exact electropherogram with when you test the exact same loci is extremely small, far less than 1 in a trillion with the current STR loci that are being used today. Okay, so let's see if this makes sense. Okay, let's see if this makes sense. And here we're looking at paternity testing, and we're just looking at one locus. Okay, so mom has on one of her chromosomes 6 repeats, and on one of her chromosomes she has 3 repeats. Dad has 5 repeats and 4 repeats. So child 1 has 6 repeats, which presumably was inherited from mom, and on child 1's other chromosome there's 5 repeats that must have come from dad. And likewise, child 2 has 1 allele from dad and 1 allele from mom, and child 3 has 1 allele from mom and 1 allele from dad. So what's going on here with child 4? Who's out there still?
So 1 allele from mom and another from the milkman.
From the milkman, exactly. That's right. Who's your daddy, right? So child 4 looks to be mom's child, but does not appear to be dad's child. And if you did a couple of other STR loci, presumably you would be able to further confirm that child 4 is not the child of this particular father. That kind of sort of makes sense?
Bob, it makes sense, but one, this seems like an abstract kind of way to approach it, and I don't know if it is more or less abstract than others. The other is, there's so many things that vary and jump around. You know, is child 4 possibly still dad's, and it, you know, other things wouldn't confirm it. It would be some sort of, you know, other.
Well, in this particular case, probably not, because child 4 has just 1 repeat and dad had 4 and 5 repeats, and the likelihood of a mutation in dad's sperm decreasing the number of repeats is extremely, extremely low. You know, you can have mutations that occur from father to child, in between father and child through the egg, I mean through the sperm, for instance. And so generally, you wouldn't rely on one locus. So this is pretty compelling, but still, to your point, you probably wouldn't rely on just one locus. You probably look at several loci to really, really, really boost your statistical power.
And they do. They do several, as you pointed out.
They do, yeah, yeah.
But okay, so this is the way it's commonly done, and not knowing it all that well, it seems like they pick something a little odd. It almost seems like, well, isn't there something more germane to how our chromosomes are us than number of repeats? I don't know. Is that really, are there other ways to look for paternity?
Yeah, you can look at SNPs, and you can look at also the variable number of tandem repeats. The thing that makes these short tandem repeats so useful, there's a couple things. Number one is they're relatively easy to isolate in the lab, you know, to cut off the edges, and they're relatively easy to measure, which is nice. The other thing that's really nice about the short tandem repeats is they happen to have considerable variability in the population. So the variable number of tandem repeats don't have nearly as much polymorphisms, and SNPs don't have nearly as much polymorphisms. You know, at a particular nucleotide in your genome, you know, some people might have an A and some people might have a G. So there's maybe 2 choices. But at each one of these short tandem repeat spots, there could be 8 or 9 or 10 different polymorphisms. And so you get a ton more statistical power with only measuring just a handful of spots because of the tremendous polymorphism that's seen with these. So it's by far the commonest method that's used, just because it's easy and it's got you know, very compelling statistics. Okay. Now we're going to get into next generation sequencing. Okay. And this will be an introduction. We're going to come back to this next week. And I probably crammed in a little bit too much information in this week, but let's try to work our way through next generation sequencing. And at a high level, the way DNA sequencing works is you are reading by writing, or you're sequencing by synthesis. And when I talk about reading by writing, what it means is if I have a piece of DNA and I want to know what the sequence is, I don't just run this through an analyzer and measure it. Really what I'm doing is I'm writing to it. And if I know that I just added a whole bunch of nucleotides and only, here I'm at this spot here where the G, and I know that only an A stuck, I can determine that only the A stuck, and by that I can infer that there must have been a G. Next, I can add a whole bunch of nucleotides, and I can see that only the G gets added. And by knowing that only the G got added, I can infer that there was a C. So I'm not exactly reading the DNA, but what I'm doing is I'm inferring the DNA by writing to the DNA and by synthesizing a complementary strand of DNA, and I know what base got incorporated at each step along the way, and from that I can infer what the sequence was in the strand that I wanted to read. So DNA testing is really reading by writing. And when Sanger, who got the Nobel Prize for coming up with this, described the process or came up with the process, what he initially was doing was he took 4 different test tubes, if you will, and he started with this same product. And in each test tube, he has all of the bases. So he's got the A, the G, the C, and the T. But he also threw in modified bases, or what are called chain terminating bases, dideoxynucleotides. And in this test tube, there is the dideoxy ATP. Then in this test tube, it's the C, the G, and the D. And these are a small quantity of each one of these are added so that periodically, as the chain is being synthesized, where an A is called for, rather than having a normal A being added, you'll wind up getting one of these dideoxy or chain terminating A's. And when one of the chain terminating A's gets added, that's it. It stops. That particular chain in that particular test tube that had the chain terminating A added is done. It's going to be fixed at that length. And so in the test tube that has the chain terminating A, at every spot where an A gets added, there's going to be some strand, some number of strands where the dideoxy chain terminating A was added rather than a normal A. And so you're going to wind up with a bunch of fragments of varying length, but at each length, that's where there was an A. And likewise, in these other test tubes, you're going to wind up with varying fragments that correspond to the addition of a chain terminating base. And if you run all of these reactions on electrophoresis, where again the small fragments go all the way to the bottom and the large fragments migrate much more slowly, you can simply say, oh, wait a minute, look at this. Look what showed up first was a C. So the smallest fragment had a C at the end, therefore the next base must have been the complement of C, which is a G. And look what came next. A G came next, so there must be a C that comes next in the strand we're reading. Then you get an A, so therefore there must have been a T, etc. So the basis of DNA sequencing is reading by writing, or the application of these chain terminating dideoxynucleotides. And in this particular case, they're running 4 separate reactions. But if you could possibly imagine each of these chain terminating dideoxynucleotides, if they could each be bound to a different colored probe, you could run this in one big reaction and then during the process of electrophoresis, just see what color comes out each time. And you've probably seen pictures like this when you're looking at the results of DNA sequencing. And really what you're seeing here is this is simply a capillary electrophoresis where the first peak you're seeing is a purple, and because it's purple, that means that it was a G, and that therefore the DNA that you're sequencing must have been a C. And then you see the next dideoxynucleotide that you see passing through the electrophoresis window is an A. Therefore, the sequence that you were reading must have been a T. So this is kind of confusing, but does that kind of sort of make sense? You're reading by writing, you're doing chain termination, and then you're looking at the different lengths of the terminated chains. So basically, you actually have a DNA molecule that was terminated at this length, and at the very tip was a fluorescent labeled dideoxynucleotide that has a G, and from that you can infer that there must be a C at this position. Kind of sort of makes sense? Okay. You guys, if you think in the feedback, let me know, for this deck, too much information, not enough information. Is this that, are we going at a good speed? Because I think this is some pretty complicated material. So if we look at, again, DNA sequencing, we can see here is the original Sanger method where he had 4 different test tubes with the 4 different radiolabeled dideoxynucleotides, and from there he could infer what the order was, or the more current technique, which is to run it all in one test tube where the dideoxynucleotides have different colors, and as they pass through the little capillary electrophoresis window, just based on the color, you can determine what dideoxynucleotide must have been added, and from there you can infer what the DNA was that was being read. Okay. Now, if you take this approach, one of the things you can do is apply this magnified enormously through what's referred to as next generation sequencing, also known as second generation sequencing or massively parallel sequencing. And in the next slide, I'm going to come back to some definitions because I think you hear these words enough: Sanger sequencing, shotgun sequencing, next generation sequencing, that the next slide will specifically come back to the vocabulary. But this is taking the Sanger sequencing method and it's applying it in a massively parallel process. So you're taking your DNA and you're chopping it up into a whole bunch of pieces, and what you're doing is each piece is in its own little well, and you're basically doing some type of modified Sanger sequencing to each piece of DNA all at the same time. So imagine, if you will, for instance, you have a slide that has a bunch of wells, and in each one of those wells you've anchored one of these fragments of DNA. So now you go ahead and add some fluorescent A, and you let it incubate, and now you look at which one of these wells light up. Aha! Well, in the well that lit up, the next sequence of that particular fragment must have been a T because it just accepted an A. Okay, now let's go ahead and add some C and see where that winds up binding. Now let's go ahead and add some G and let's see where that winds up binding. And so you're still doing a type of Sanger sequencing, but you're doing it in a massively parallel fashion. You are applying that sequencing to a boatload of fragments all at the same time. And what you wind up with are a whole bunch of these short reads of DNA sequence that now you know exactly what the DNA sequence is of each of these reads. And what happens now in next generation sequencing is each one of these reads gets aligned or mapped to a reference DNA. So you grab the reference genome from the NCI site, from the NCBI site, and each read is mapped. And what winds up happening is each of these fragments, depending on the technology, let's just say each of these fragments is approximately 300 bases long, and in any particular spot in the any particular spot in the genome, you'll typically have somewhere between, say, 50 to 500 reads. Here, it's showing you that there were 10 fragments overlapping this particular section of the reference genome, and there was 5 fragments overlapping this particular segment of the reference genome. And the more reads that you have overlapping a particular area of the reference genome, the greater accuracy you have to say exactly what the base was there. So really what you do is you fragment the DNA, you do a whole bunch of short reads, maybe millions of these short reads. You are aligning them all to a reference genome, and having done that, you then can determine exactly what the DNA sequence was. One of the things you see here in this picture is that you have these 10 reads, but for half of the reads, we find a G at this spot, and for half of the reads, we see an A at this spot. So what could be going on here? The reference DNA has an A at this spot, and half of our reads we're seeing an A, but half of our reads we're seeing a G. What do you think could be going on here? So keep in mind that this is the DNA that you're analyzing from the patient is the DNA from 2 chromosomes, right? You got chromosome 7 from mom, and you got chromosome 7 from dad. And what you're seeing here is this particular patient is heterozygote at this particular spot, because half of the reads are showing a G at this spot, and half of the reads are showing an A at this spot. So from there, you can infer, oh, okay, well, on one of the alleles from this patient, there's a G at that spot, and one of the alleles from this patient, you're seeing an A, and this is what you would expect to see if the patient is heterozygote for this particular locus. That kind of sort of makes sense? Okay. So the vocabulary, this particular vocabulary is not covered in the sequence ontology, but it also varies quite a bit depending on where you look. So we'll see Sanger sequencing, shotgun sequencing, next generation sequencing, third generation sequencing, massively parallel sequencing. And so for the most part, these definitions are pretty good, but the definitions of third and fourth generation sequencing probably have the greatest variability that are out there. Sanger sequencing is the basic process of reading by writing with chain terminating nucleotides. Shotgun sequencing is this process where you break the DNA up into fragments, sequence the fragments, then map each fragment back to a reference sequence. Next generation sequencing is generally synonymous with massively parallel sequencing, like we just looked at on the prior slide, where you're breaking the DNA up into fragments and sequencing many fragments in parallel. And for the most part, when people talk about third generation sequencing, what they're talking about is very long reads. And so if you look at today's next generation sequencing technology, these reads, these fragments are only about 300 bases long. And as we'll cover in more detail next week, these short reads can pose considerable challenges when trying to map them to the reference genome, particularly when you recall that our DNA has 50% of it is repeats. And so if 50% of our DNA is repeats, then you can imagine that there's going to be a lot of these fragments that don't map unambiguously to just a single spot on the reference genome. And so if we had technology that allowed us to have reads not just of 300 bases, but of 10,000 bases, then we could much more easily align those, map those to the reference genome, and a lot of the quality and ambiguities, the quality issues and the ambiguities that we're facing today, that we'll cover in a lot more detail next week, could be addressed. So third generation sequencing, very long reads, up to 10 to the fifth bases. And fourth generation sequencing also has considerable variability in the definitions, but is oftentimes making reference to in situ or single cell sequencing. So for instance, if you take a tumor specimen, you're not just crunching up all the DNA and analyzing it as a whole, but really you're looking at the DNA of this particular cell, and you're looking at the DNA of this particular cell and of this particular cell, and that allows you, for instance, to track the lineage of mutations in that tumor. And then I think, oh my gosh, I'm going to have to speed up a little bit. Is this a good speed for you guys? Or is this too slow, too fast? What do you think?
I think it's good.
There? Good? Okay.
It's good.
Okay. All right. It's going to be a little bit tight. I put in a lot of information for this week. This is an example of third generation sequencing. And by third generation sequencing, I mean people trying to do very long reads. Some people would refer to the nanopore technology as fourth generation. And their definition of fourth generation is you're reading directly, you're not reading by writing. But what characterizes both of these is extremely long read lengths. They're both being used in research, but neither of these are being used for any clinical applications right now. And so these are just some examples of coming technology where you can achieve very long read lengths, 100,000 bases in a row. The PacBio system passes the DNA through the DNA polymerase. The DNA polymerase is actually fixed in the well, and as the DNA passes by it and the base gets added, each base, as it's incorporated, gives off a little fluorescent signal, which is then detected. And in the Oxford nanopore method, the DNA itself is just passing through the nanopore, and each base, as it passes through, gives a slightly different electrical signal. And so some people would say that the nanopore technology is fourth generation because it's direct reading. It differs from the others in that it's not really using the Sanger D-deoxy method. One of the applications of DNA sequencing is in prenatal testing. And it turns out that our serum contains cell-free DNA. We all have cell-free DNA circulating. I think we may have talked a little bit about looking at cell-free DNA for cancer screening, because it turns out that cancer cells shed DNA. It also turns out that when a woman is pregnant, she has a small amount of fetal DNA circulating free in her serum. So cell-free fetal DNA And one of the techniques or methods that's being illustrated here is that if you sequence a sample of cell-free DNA in maternal serum, and you take each one of these fragments and you map them to the corresponding chromosomes, what happens is if you see that there's more fragments corresponding to, say, chromosome 22 than would be expected, then that from there you can infer there must be extra chromosome 22. And it turns out to be a very reliable test for Down syndrome for trisomy 21, in that by just taking the cell-free DNA in maternal serum and mapping the fragments back to the chromosomes and seeing, you would expect to see a certain number of sequences or fragments per chromosome relative to the size of that chromosome. And if you see a statistically increased number of fragments mapping to chromosome 21, it tells you that there's extra copies of chromosome 21. And from there, and this turns out to be a very reliable screening test for Down syndrome. This slide here is a summary of the various testing mechanisms. I just think I'm way off on time, and I don't want to rush through this stuff. I may let some of this material spill over into next week. Yeah, I'm just I think this material is going a little bit longer than I had anticipated. So here's a summary. So we see this is a patient obviously with trisomy 21, and here we see the karyotype where we simply see 3 chromosome 21s. Here is the results of a FISH test where we see the red is a chromosome 21 probe, and the green is a control autosome probe. So we see 2 greens and 3 reds. And then what we see here in portion C would be one of these comparative genome hybridization arrays where we would have mixed patient DNA with control DNA, and we would see that there's looks like extra copies of chromosome 21. And then at the bottom, we see whole genome sequencing where you're just seeing a lot more reads from chromosome 21. And this might be kind of sort of similar to the fetal DNA testing. You're doing next generation sequencing, and you'd expect to see an even number of reads across the genome, but you're seeing a lot more reads corresponding to chromosome 21. So let's take a look at how this information might be communicated. Okay, we've looked at the HL7 version 2 molecular reporting, the LRI ballot. We haven't looked at the HL7 version 2 cytogenetics ballot. If I were to say how these differ, and these are both, the LRI ballot, the LRI standard is in ballot, whereas the cytogenetics standard passed back in 2014, and they're both active. There's no intent for the LRI standard to subsume or trump the cytogenetics standard. And so you can kind of sort of think of these as these are both intended for labs reporting to electronic health records, and if the lab is reporting molecular results, next generation sequencing or targeted sequencing, then the LRI standard would be applicable, whereas if the lab is reporting the results of cytogenetics, then this particular standard would be applicable. And by cytogenetics, what's encompassed in this standard are the karyotypes, the FISH, and the chromosomal microarrays. So if you're trying to communicate karyotype, fish, or chromosomal microarray information, this would be the particular standard to use. And there's a more complete HL7 version 2 example that I put on the download site if you're interested. But just to give you an idea of how how these might look, the karyotype, the fish results, and the comparative genome hybridization array might look when they're communicated in an HL7 message. The karyotype might look as a 47XY plus 21. And one of the things to note here is we're now using a different syntax. We've talked about using HGVS syntax to communicate simple variants. But when it comes to cytogenetics, labs report cytogenetic results using a different syntax called ISCN. And ISCN really was kind of designed, I guess, from the top down, and HGVS was kind of designed from the bottom up. You know, HGVS was really designed to communicate molecular results, and ISCN was really designed to communicate the more macroscopic results. They're meeting in the middle because a lot of these tests that previously had been communicated using ISCN syntax are now able to be performed using molecular DNA technique. And so ISCN and HGVS are kind of working on collaborating going forward. But for now, you'll often see labs. In fact, labs are required to report ISCN syntax for cytogenetic findings. And so what you might see, for instance, for a karyotype would be 47XY plus 21 for Down syndrome. When you look at the results coming out of fish, what you'll see is 3 probes were applied in this case, and in each case, 3 copies of the probe showed up. Now, one of the things you'll notice here is that you're inferring trisomy 21 because you're seeing, now these probes are all probes that are in the tail end of chromosome 21. They're down at Q22, or the very tip of Q22, which is felt to be the critical region for Down syndrome. It's felt to be the critical region for Down syndrome because mutations there have, or at least segmental deletions in that region, result in most of the phenotypic features of Down syndrome. So here you have 3 probes that are specifically targeting the Q22 region of chromosome 21, and they're all present in triplicate. And so from there, you can infer that this patient has Down syndrome, even though you haven't necessarily seen that, even though you don't know for sure based on this that there's complete trisomy 21. You just know that this particular region of the chromosome, there's a 3rd copy based on fish. And microarray is kind of similar in that it's looking at targeted regions of the genome, and the microarray results are also dependent on what particular probes were being used in the microarray array. So here, for instance, you see that in chromosome 21, in region Q22, there's a large area in this particular reference genome from 35,900,000 to 48,100,000. There's a 3rd copy. Okay. So again, the microarray isn't necessarily conclusive that there is trisomy 21. You know, 95% of the time when you have Down syndrome, you have trisomy 21. So it's a pretty good guess that when you see these results in a microarray, you have trisomy 21. But really what the microarray specifically is telling you is that this specific region of the chromosome, we're seeing that there's a 3rd copy of it. Okay, and it might be that you want to go back and do karyotype as a way to really confirm that this is trisomy 21. Okay.
Bob? I mean, I'm
Yeah.
sure there's some that are partial, the other 5%, but wouldn't every person who had 3 copies of this be a Down? And there's some that don't have the full 3 copies, but you're saying it, when you're saying it, it sounds like some people who have triplicate for this large region wouldn't have Down syndrome. Is that
So
correct?
everybody would presumably have Down syndrome, but not everyone would necessarily have trisomy 21. Right? They could simply have a segmental duplication.
Right, right,
Right.
right.
So
But
so
everybody
so,
has-
so yes,
everybody
they
has-
would have.
would have Down. There are another 5% that are segmental.
Right. That's right. That's right. So only here on the karyotype did you really know that it's trisomy 21. From FISH and from microarray, you know that it's you can say Down syndrome, and you can guess it's probably trisomy 21,
Oh,
but you
I see
can't
what you're
say for sure. Yeah.
Okay.
Okay.
They have Down syndrome, but okay, they may not have a full trisomy. They may have partial. Okay.
That's right. That's right. Okay. All right. All right. Okay. Quiz questions. A paternity test is looking for matches in which type of polymorphism? One C. Short tandem repeat. Did someone say C?
C.
Good. Okay. What type of test might you order to look for a translocation?
Karyotype.
That's a yeah, and this is a tough question because if the translocation is big enough, then you could very well see it on the karyotype. If it's small enough, you might miss it on karyotype. You might miss it on FISH. You might only pick it up on full DNA sequencing. And so, you can do karyotype, you can do FISH. If you're strongly suspicious, it might be that you have to do some different type of test, right? If you suspect a translocation and you do a karyotype and you don't see it, then if you want to drill down to the next layer of resolution, then you could do FISH, for instance. Let's see. We probably have another 30 minutes of information. I'd kind of like to crank through it. So I wonder how you guys feel about that. If we go over 5-10 minutes today, anyone have any strong objections to that? I'll have to try to better gauge how much material to put in. I tell you, there's just so much stuff. I mean, and like we're scratching the surface with material. I'm gonna plan on maybe running 5-10 minutes over today, and I guess if you have to drop off, you can pick up the video and listen to the last 10 minutes later. Is what we'll wind up doing today. And thank you guys again for bearing with me on this kind of dry run. The feedback I've been getting has been just super helpful. So let's switch gears now. We're going to look at a clinical case, and based on this clinical case, we're going to then come back to DNA testing and try to figure out exactly what test we want to order. So this is a guy, a 37-year-old guy. He was admitted to the hospital in 2001 with shortness of breath and confusion. And he was found to have both pneumocystis carinii, pneumonia, and Toxoplasma gondii encephalitis. And that led to him being diagnosed with AIDS. And at the time, his CD4 count was 2, and his HIV-1 viral load was 120,000 copies per mil. So he was started with some medications, some trimethoprim sulfa, and he was started on antiretroviral therapy that included a drug called abacavir. Okay, so this guy was started on abacavir, and his encephalitis and his pneumonia cleared, and he was discharged from the hospital on oral therapy. And 2 weeks after beginning his antiretroviral therapy, he came in with a non-febrile generalized rash. It was on his palms, his mouth. His blood pressure was 130/60. His temperature was 37.1. His pulse was 88. He was breathing 15 breaths per minute. His oxygen saturation was 96% on room air. And he had this rash, this disseminated cutaneous eruption with these discrete dark red macules covering 90% of his body surface area. There was roughly 5% of his epidermis showed detachment. He had genital ulcers. He had ulcers in his mouth. He had ulcers. He had lesions in his conjunctiva. And minor pressure on his skin resulted in some sloughing of the skin. So he was diagnosed with what's called Stevens-Johnson syndrome, a hypersensitivity reaction, and skin biopsy was compatible with Stevens-Johnson syndrome. And because back in 2001 they had seen some reports that there might be some cutaneous hypersensitivity reactions with abacavir, the drug was stopped. He was transferred to a burn unit, monitored, and he began to heal over the next week. And within 3 weeks, his skin lesions completely resolved. His abacavir was changed to different antiretroviral treatments, and without recurrence of the skin reaction, and his viral load decreased to undetectable, and his CD4 count returned to normal. And a year later, when the increased susceptibility to Stevens-Johnson syndrome with abacavir was shown to depend on a particular genotype, HLA genotype, he had HLA typing and was found to carry the susceptibility allele, which is it's an HLA-B allele called 5701. So this guy was found to have the HLA-B 5701 allele. And so that kind of leads us into this notion of pharmacogenomics and drug-gene interactions. And I want to talk a little bit about this particular case, but I also want to digress a little bit and talk about pharmacogenomics kind of at a high level. For abacavir in particular, what we see, again, is the HLA-B gene. The HLA-B gene results in this HLA molecule sitting on the surface of a number of cells, and this molecule presents antigenic peptides to white blood cells as a way of triggering an immune response. And it's thought that abacavir can interact with the HLA-B molecule, specifically with one particular allele of this molecule, the 5701 allele. It can interact with this binding groove, and it then alters the binding properties of that particular molecule, allowing it to bind and present novel peptides that it wouldn't normally have presented. And that then results in the HLA molecule presenting novel peptides and triggering a hypersensitivity reaction. And that's specifically for the HLA-B 5701 allele. But if we back up a minute, let's just digress for a couple of slides and talk about pharmacogenomics. Really, when people think about pharmacogenomics, they're kind of sort of breaking it down into three categories. These are genetic variants that affect one of these three categories, and that includes drug metabolism, drug efficacy, or adverse drug reactions. Excuse me. when we talk about drug metabolism, what we're really talking about is drug levels. And so there's certain genetic variations that will give you a higher drug level or a lower drug level. One example is codeine and its interaction with a particular cytochrome P450 gene. So pharmacogenetics can affect drug metabolism, in other words, drug levels. Pharmacogenomics can also affect drug efficacy, or in other words, what is the effect of a drug at a given level. So two people can have the exact same level of a drug, but in one person, that drug may be toxic, and another person, the drug may just be pharmacologic. And an example of that might be the succinylcholine and the plasmacholinesterase gene. And for this particular case, there's an FDA label warning that individuals who are carriers of an atypical variant of this particular gene are at risk of prolonged apnea if they're administered succinylcholine. So again, it's not changing the drug level, it's changing the drug efficacy. And then finally, genetic variants can also affect adverse drug reactions, put you at risk for adverse drug reactions, such as immune-mediated reactions, such as we're seeing in this particular case with abacavir and HLA-B. I'm going to digress just a little bit more for the drug metabolism case, only because it's talked about so much that I wanted to at least introduce the topic just so you could see a little bit about it. And then we'll come back to abacavir. So again, we're talking now about how genetic variants affect drug metabolism. The abacavir case really is an example of how a genetic variant increases the risk of an adverse drug reaction. But if we look at drug metabolism or pharmacokinetics and how genetic variants affect pharmacokinetics, a real common example you'll see out there are changes in the cytochrome P450 enzymes. And there's a couple things to note here. So let's see, cytochrome P450 is kind of a broad category. There's 56 different cytochrome P450 enzymes. Each one of those enzymes is from a different gene. Okay, so there's 56 different cytochrome P450 genes. They're highly polymorphic. They group these genes or the enzymes into families, and that's largely based on amino acid homology. And so when you look at the naming for a particular gene, in this case CYP2D6, you can kind of parse that a little bit. The 2 represents the family, the D represents the subfamily, the 6 represents the isoenzyme. So they've broken down these 56 genes into 20 families, but really out of all of the 56 genes, there are 6 of them that metabolize more than 90% of commonly used drugs. And so what you see in this figure here are the 6 cytochrome P450 genes, or if you will, the cytochrome P450 enzymes that are responsible for metabolizing greater than 90% of commonly used drugs. The other thing to notice here is they have this kind of star notation for indicating the alleles. And you may remember that we saw this when we looked at HLA. It's relevant because it's a big deal for pharmacogenomics. Sometimes you'll see when you're looking at the results of a variant, you'll see HDVS notation. You'll see that this particular gene at this particular spot varied from the reference genome. With the cytochrome P450 genes, it might be that the variant you have differs from the reference genome, not just at one spot, but maybe at 4 spots. And at one spot, it's an inversion, and at one spot it's a deletion, and at one spot it's a SNP change. And in order to simplify the communication of exactly what cytochrome P450 allele you have, they've simply named the alleles. And so allele number 4 of this gene, you can go to a mapping table and you can figure out exactly what all of the molecular level variants are. But for convenience, there are certain genes where they have such a degree of polymorphism that it's common to name the alleles as opposed to naming the individual HGVS level variants. Okay, and we'll come back to that because it's important when we're communicating the results of alleles, and it's also important when, for instance, a lab sends you the results of a molecular analysis in HGVS syntax, and yet your pharmacogenomic decision support is triggered off of the allele. There's a need to be able to map between alleles and the molecular HGVS type syntax. The cytochrome P450 enzyme system is responsible for pharmacokinetics, and based on the particular variants that you have, you might see that the individual is a poor metabolizer of a given drug, a normal metabolizer, or an ultra-fast metabolizer. Now, this can go one of two ways depending on whether the drug that's being administered is the drug or is a prodrug. So for instance, with codeine, codeine is weak, but codeine is converted to morphine, which is strong, and the conversion requires the cytochrome P2D6 enzyme. And when you have a poor metabolizer, okay, when you have an allele of this particular gene that means that you are a poor metabolizer of codeine, you'll simply have very little therapeutic effect from codeine. And so oftentimes poor metabolizers aren't given codeine because the codeine is just not going to work. On the other hand, you also have problems with ultra-fast metabolizers because if an ultra-fast metabolizer is given codeine, they're going to metabolize that codeine to morphine so quickly that they can become intoxicated even with low doses of codeine. And so it's important not only to understand what the pharmacokinetics are based on whether or not you are a poor, a normal, or ultra-fast metabolizer, but it also has to be kept in mind whether or not you're administering the drug or the prodrug. Okay? And I think with that, I'm going to start weaving my way back to abacavir to finish up, although I will indicate that there's a growing list of gene-drug interactions, and I'm going to take you to a website in a minute where you have an up-to-date list. But here's an example of genes and drugs that are cited in FDA package inserts. And not only will they cite that there's an interaction, but they'll cite recommendations. So let's take a look at some of the bioinformatics aspects. And what I've shown you here is a basic pharmacogenomics clinical decision support rule. And this is the type of rule, for instance, that might be applied at order entry. And it might be applied to any drug. Okay, it's kind of a general rule. And that rule would be, okay, you've just tried to order a drug, and is there an interacting genotype? If not, okay, you're all done. But if so, do you have a genotype on file? If you do, then does that genotype recommend that you change to a different drug? Does it recommend that you change to a different dose? If you don't have a genotype on file, then do your guidelines recommend that you need to order a genetic test up front? And if so, what test would you actually order? And then finally, you might want to provide information on drug-gene interaction. So let's go through this decision support scenario in a little bit more detail, because it turns out that the information for answering these is generally available, although not necessarily always in complete computable structured form, although that is coming along pretty nicely too. So drug order attempted, is there an interacting phenotype? And the GKB website is nice in that it gives you a list of drugs and interacting genotypes. And so, for instance, in this decision support rule, if there is a drug on this list, then the answer to the question is, yes. There is, is there an interacting genotype? Well, if you see it on this particular list, then yes, abacavir is associated with HLA-B. Now the question is, if a drug order is attempted, is there a genotype on file? And here's where things get a little bit tricky, and this is kind of a fun or not unfamiliar informatics challenge. How many ways can you say HLA-B5701 positive? There could be a generic test where the LOINC code for the test order is HLA-B, and the result is allele 5701. Or there could be a LOINC code where the test is HLA-B5701, and the result could be positive. Or a lab might just send you back in HGVS notation what the particular variants were from which you could glean that this corresponds to the 5701 allele. So in an EHR, there's going to be a lot of different ways in which you can assert B5701 is positive, and so that needs to be kept in mind when answering this question, do I have a genotype on file? If there's a genotype on file, then the question is, do we need to change to a different drug? And here for abacavir, if we go to the drug label, what we'll see is from the FDA, the FDA-approved label for abacavir states that genetic testing for this allele is required prior to initiating, and it says abacavir is contraindicated in patients with this allele due to the risk for hypersensitivity. So it says, if you know that they have B5701, don't use this drug. And it says, if you don't know whether or not they have B5701, you need to test for it. You need to test for it before administering the drug. And these guidelines are kind of semi-structured. There's another group called CPIC, and there's a link to CPIC in the deck. CPIC is working with this particular site, PharmGKB, and what they're trying to do is take a lot of these guidelines and create EHR-ready structured clinical decision support tables. And they have these decision support tables available for a subset of these interactions. And so down the road, it might be that using the CPIC resources will be immediately plug and playable for clinical decision support. But as it stands, there's a little bit of massage work needed to be done. The data that's necessary for decision support is available on this particular site, but it's not necessarily directly amenable for immediate plug and play in an EHR. So if there's a genotype on file, should you change to a different drug? And again, per the FDA, yes, abacavir is contraindicated. And if a drug order is attempted and there's no genotype on file, in this particular case, do you need to order a genetic test? Yes, it would be ordered. And then the question is, what genetic test should I order? And just as an example, we have here a website, also at the NCBI, a genetic testing registry. In this case, I plugged in the SNOMED code for a Abacavir allergy, and I'll just cut to the chase and say we didn't get any results. So what we can do is we can search for Abacavir, and we'll wind up finding that there's 6 tests available. And if we look at the particular tests, let's just look at the ones in the United States, we'll find that there are 4 tests that have been registered. And by the way, the genetic testing registry at the NCBI is a voluntary registration of tests. So anyone who in the U.S., such as Arup Labs, that has a test can register that test here in the registry. And if you look at these tests, most of what these tests are doing are PCR with allele-specific hybridization. Okay, so PCR with allele-specific hybridization. And let's come back to that, because at this point I think it's going to be clear to you now what PCR with allele-specific hybridization is. But before we do that, let's just walk through this scenario a little bit and look at some of the bioinformatics barriers. These particular sites right now are not indexed by RxNorm. The GTR site is indexed by SNOMED code, although, as we saw for Abacavir hypersensitivity, in that particular case, we couldn't find it by SNOMED code. So right now, when searching, and PharmGKB has announced that they are going to support RxNorm-based searching, but they don't today. So right now, to interact with these sites, it's a bit of a manual process. There are multiple allele representations, and so if you want to do pharmacogenomic clinical decision support, you have to come up with a mechanism for, aligning the multiple allele representations. The test results, when you identify tests in GTR, even though there's a field in there where the submitter can include a LOIN code, at least for the ones that I looked at, I didn't find any LOIN codes. And so, to me, an ideal scenario would be you identify test results, but then you align those back to orderables in your EHR to just enable the clinician to click on the particular order. But these codes, the GTR resource generally doesn't have LOIN codes, and so the process of identifying a test in GTR and then figuring out exactly what then you would need to order in your EHR also remains a bit of a challenge. And the CDS components are not fully structured, and the CPIC is addressing this, and there's a growing number of these PharmGKB resources that are structured, but there's a boatload of drugs that have genotype information, and there's a boatload of FDA package inserts for which there's not currently completely structured information amenable to just decision support in EHR, and that's still evolving as well. So if we look at that, now let's come back to this test that we said we wanted to do, okay? So we said we wanted to do PCR with allele-specific hybridization, and I think now maybe this will make a little bit more sense. This isn't necessarily exactly the mechanism by which the particular lab would be doing the test, but an example of an allele-specific probe might be, okay, I have a slide and I include a probe for the normal sequence, and I include a probe for this particular variant. I'm going to then incubate the patient's DNA, and I'm going to see, does the patient's DNA hybridize with the normal probe? In this case, it did not, or is the patient going to hybridize with the mutated sequence? Now you can imagine I could put in a probe for particular HLA-B5701 allele. And I could put in fact, I could put in probes for all of the known HLA-B alleles, and we could see, based on where hybridization takes place, which particular allele the patient has. And so this is kind of at a high level conceptually, when the lab is saying it's doing PCR with an allele-specific probe, it's doing something like this. It's hybridizing the patient's DNA against a set of probes to try to find where the hybridization is taking place. And finally, oh my gosh, you guys have bared with me for so long. I appreciate it. Finally, we want to communicate the information. And in the May 2017 LRI ballot, we've talked a lot about communicating discrete genetic variants. There's also a section in that ballot designed for communicating pharmacogenomics genotype panels. And if you looked at this back in earlier drafts, it's been modified now. This particular section addresses 3 use cases. It addresses the use case where the genotype affects the pharmacokinetics. There's a use case where the genotype affects the drug efficacy. And then the third use case is where the genotype affects your risk for an adverse reaction. So the three subtypes of pharmacogenetic information that you can convey now in the standard relate back to, is there a drug metabolism issue, is there a drug efficacy issue, or is there an adverse drug reaction risk? Okay. And an example in this case for abacavir of how this information might look is the gene being studied is HLA-B, and here's the genotype. And so one of the patient's alleles was 5701, and one of their alleles was allele 35, which is a benign allele. But the presence of even a heterozygote allele 5701 is known to put the patient at high risk, and so the variations affect on high-risk allele if they're high risk, and this would be for abacavir with a recommendation of consider alternative medications. And so this evolving standard allows for communication of pharmacogenomics. In this particular case is an example of risk, but you could also, using different fields in the standard, communicate the effect on the drug kinetics. Are you a fast metabolizer or slow metabolizer? And also you could communicate drug efficacy. Is the drug going to have higher action? Is it going to be less active? And finally,
Bob,
in order to...
Eric, can I ask one question?
Yeah.
Go back to that previous slide where you showed for allergies or contraindications, is there anything that gets a little more specific than consider alternate medication? Is there levels of risk that are assessed at some things? For example, when patients say they're penicillin allergic, a lot of times cephalosporins are thought to be contraindicated, but it's a relative contraindication, and most of the time we just ignore it.
Right.
And
right,
even
right.
so, those penicillin allergies, if you say, well, I had a rash, and we can ignore most of those too. So
Yeah.
just consider, is there anything, I think that we're getting down to genetics, it ought to be a little more information than consider.
So a couple things. One is, there are separate HL7 messages for communicating allergy information. If you're allergic to penicillin or what have you. This particular message was designed specifically for pharmacogenomics. And so its intent isn't necessarily to say, you know, that you have an allergy to abacavir. I mean, you could, but this was designed to say, because you have this genetic variant, you'd be at high risk if given abacavir. It's not designed to be communicating allergy information per se. That said, this is still going through ballot, and so while it doesn't allow you right now to expand on the details the way you're talking about, it's possible that in the final iteration, before it becomes a standard, that might be fleshed out.
If anybody's working on that, I would certainly say this is, I know from looking at information on what people often ignore allergy stuff, sometimes to their detriment, but often not. And I would say the same thing is true here. Unless there's some level of alert, it's going to not be as useful. And I would imagine we would know that better than this.
Right, right. Okay, so in gratitude for you guys sticking with me for so long, you get in exchange, you get to be the ones to answer the quiz questions. Okay, that's just my little way of saying thank you. Okay, so number one, genetic variation can result in A, B, or C.
D, all of the above.
D, all of the above. Very good. Okay, number two, assume that drug X is a prodrug, and you can think of codeine as drug X. It's a prodrug. It's converted to an active form after administration. Okay? So what would you anticipate if the patient is a slow metabolizer? How effective is drug X going to be?
Less effective.
Less effective, that's right. It's going to take a very long time to convert the prodrug into the active form. What if the patient is an ultra-fast metabolizer?
Toxic.
Could be toxic, that's right. That prodrug is going to be rapidly converted to an active form, and they could become quickly toxic. Very, very good. All right, guys. Thank you very much for bearing with me. Sorry we ran over. If you do have any, if you are not rushing off and you do have any feedback for today's session, I can stick around. But the presentation's over anyhow. See if I can turn off the recording real quick.
Thank you, Bob. I thought it was really good and well paced for me, and very clear and understandable.
Thank you. Thank you. Alrighty, guys. Well, thank you guys very much for coming, and I look forward to next generation sequencing next week with you.
Thank you.
Okay. Bye, guys.